All right, so we are recording. So welcome to study session for exam 3. We're going to go over module 10, 11, 12, and 13. So each of these modules goes more into evolution, biodiversity, so on and so forth, talking briefly about the different kingdoms, domains. So we have like our animal kingdom, plant kingdom, we have our different domains with bacteria, archaea, eukaryotes, and yeah, just going into each of those. So first off, talking about module 10, I have the learning objectives listed. Oh, did the screen close? Oh, sorry, my computer is going crazy. Can anyone still see the iPad screen? Okay, it's a black screen. Okay, now it's back. Okay, good. So now you can see it. Okay, so module 10 is talking about Darwin's theory of evolution, how it differs from present day theory. We're talking about the 4 basic causes of evolution, the definition of species, how to identify them, how they are different from one another, and also looking into common misconceptions that people have about evolution in general. So going into Darwin's theory of evolution, so Darwin was a scientist that went to the Galapagos Islands, I believe in the 1800s. And when he was in the Galapagos Islands, he looked at different organisms such as finches. So actually here is the picture that he had identifying different finches. And he noticed that each of these different types of finches had different traits to each of them, even though they were all technically finches. So you notice with the first one, they have a very broad beak. It's very big compared to the second, third, and fourth one. And you notice that their coloration can be different, their eye size can be different. So basically, he determined that all species of organisms arise and develop through natural selection of inherited variations that increase the individual's ability to compete, survive, and reproduce. So basically what's happening with each of these different finches is in their environment, they have different types of niches that they have to have in order to survive. So in the case of the first finch, they have a larger, broader beak. So maybe the type of food that they eat involves them having to have a larger beak, whereas the fourth finch, it has a lot smaller of a beak. So maybe it just eats like berries or softer foods that wouldn't need to be broken up. So because of their different traits, they're able to compete, survive, and reproduce in their specific environment. So that's kind of basically what Darwin proposed for the theory of evolution. And this theory does describe facts about the living world and kind of how organisms have evolved to where they are now. So going back to natural selection is basically talking about organisms adapted to their environment. They tend to survive and produce more offspring. So I have a little diagram here of giraffes. I know it's a really bad picture, but imagine those are giraffes. And the 2 giraffes here with the long necks, you notice that they can reach the top of the tree, whereas that giraffe on the very bottom, it does not have a long neck, so it won't be able to reach those leaves, it won't be able to eat anything, it's probably going to die because it doesn't have that long neck. Also, on top of that, giraffes, when they are looking for a mate, they have fights, like neck fights. I don't remember the specific name, but if you look it up, it's hilarious. Definitely look into it. Basically, in order to compete for mates, male giraffes will come at each other and hit each other with their necks. So having a longer, stronger neck also helps them attract mates, and thus they're going to create more offspring. So because of natural selection, the giraffes with the longer necks are going to pass on their trait to their offspring so they can survive better. And basically natural selection is really depending on the phenotype. So if we remember from the last exam, the last few modules before this one, the phenotype is basically a trait that you can see on the outside. So in this case, you can see that they have a long neck. Or in the case of the finches, you can see that each of them has a different beak size. So according to their phenotype, that's what gives that individual an advantage, or it could even give a disadvantage in the struggle to survive and reproduce. So in the case of the giraffe at the very bottom, they would have a disadvantage for having a small neck because they would not be able to survive eating off of these tall trees and they would not attract mates. All right, so now going into species. Species are a group of actual or potentially interbreeding individuals. So basically, these are organisms that are capable of producing offspring with one another. So if two organisms are able to produce offspring, they're usually within the same species, but of course there are some exceptions where in her video for the module, Dr. Ortiz talks about hybrid animals. So there are cases where hybrid animals can be produced that may be advantageous, that could produce an offspring that is not, like they have the disadvantage in their environment. So that would be unfortunate. Oh my gosh. Okay, screen is back. Yeah, so those are different species, and different species, this is when we're focusing more on the genotypes. So species have a different genetic makeup from one another. So in this example here, where we have two different birds, these are meadowlarks. They are different species because they have a different genetic makeup, but they do look somewhat similar. But in the case of the gaudy commodore, which is a type of butterfly, they're the same species even though they look very different from one another. And that's because their genetic makeup is more similar and they can pass on traits by producing offspring. Are there any questions so far? If so, I just want to remind you guys, if you have any questions throughout the study session, feel free to type in the chat or unmute yourself. That's fine with me. All right, so I'm going to move forward into speciation. So speciation is the formation of two species from one original species. And there's two types of speciation. There's allopatric and sympatric. So allopatric is speciation in another place. So basically, if you have two different species, it's because they were separated. So I actually have this diagram here that shows the difference between the two types of speciation. So as you can see in the allopatric scenario, there's a mountain range that is now in between the land that had all of these pine trees. But because of this separation in the land, now you have one species of trees on one side and one species of trees on the other. So this has really good so this has genetic material transfer from one population to the other. Or actually, I'm sorry. This one would have less gene flow because gene flow is basically transferring material easily. So it's difficult for genes to travel over all of those mountains to the next type of tree. And then we have sympatric speciation. So this is speciation occurring in the same place. And so it occurs with the original species in one location. So what could happen in this case is polyploidy could happen. There could be an error in meiosis, and now a new species is going to come about. There could be a difference in sexual selection. So mates may prefer to select for specific characteristics. This is not really, a tree's not really a good example with this one, but let's say that you have two birds living in a certain area, and usually birds, when they look for mates, they look for a male that has very bright colors or extra feathers. So in this case, because of those specific characteristics, they could be selected. So that would be sexual selection. And then resource differentiation. So this is a competition of resources. So let's say that one resource is really abundant, that species may come about. There will be a lot more of that species than the species that is affected that has little resources. So those are drivers of sympatric speciation. I would definitely know those for the exam. And then the module also talks a bit about mutations. So mutations in genes can put organisms at an advantage or a disadvantage. So a really good example of this is with antibiotic resistance mutation in bacteria. So this is actually a really neat video that you can find on YouTube, on the interwebs. If you look up Harvard Medical School, I believe they did this experiment, yes. And so basically what they did was they had a huge Petri dish. It was like the size of an entire table. And usually Petri dishes are used to grow bacteria. They have all the nutrients for bacteria, so on and so forth. And so this first picture right here with all the numbers, this is the big Petri dish, but it's divided into sections. So with each section, they added antibiotics, and antibiotics actually are used to kill bacteria. So as the Petri dish is going from each side to the middle, more and more antibiotic concentration is put onto the plate. So at first, the bacteria is put onto the Petri dish, and you can see on the sides, like there's that really dense, it's like white mark on the Petri dish that represents bacteria colonies. So you can see like they grew super easily in that first part where there was no antibiotic placed on the plate. But as they went to that next section with some antibiotic, you can see there's these kind of clouds coming off, and that's because of mutation. So basically the bacteria mutated so that it would resist the antibiotic. So slowly and slowly, each bacteria eventually mutates to the point where it can resist that antibiotic concentration in the middle, which is 1,000 times more than on the outer edges. So really crazy, in this case, mutations were advantageous to the bacteria because now they can survive that colony. The colonies that you find in the middle, those would be able to survive against antibiotics. And then briefly talking about genetic drift. So this is a change in alleles frequency due to chance. So a good example of this is usually using the bottleneck effect. So basically, let's say you have animals in a certain environment and a natural disaster occurs. So completely randomly, you are going to have this huge population of animals all of a sudden bottleneck down into as few that are left. And so completely randomly, due to genetic drift by chance, one species would prevail while the others would not. So in this case, the red frogs died off, but randomly, the green frogs ended up surviving. Yes, so that is genetic drift. And then lastly, in this module, we cover evolution misconceptions and criticisms. So these are all misconceptions. These are all not true. I would definitely know all of these. So evolution is not just a theory. As I said before, it does describe, high up there. This theory describes facts about the living world. Oh no. There it is, sorry. An individual does not change as it ages. Actually, individuals do change as they age. This is referred to as development. Evolution explains how life originated. It does not explain how life originated, but more so how animals or plants or all types of organisms have obtained new traits as time has gone by. And organisms evolve as a response to a change in an environment. That is not true as well. So these are all misconceptions and criticisms. And like I said before, it's really handy to know those for the exam. So that's pretty much module 10. Are there any questions before moving forward? Okay. So we'll move on to module 11. So module 11, we're talking about taxonomic classification, systematics, taxonomy, relate to phylogeny. We're talking about phylogenetic trees, very briefly, cladistics, cladograms, like you did in your assignments, and comparing homologous and analogous traits. So first off, the levels of taxonomic classification, definitely know the order of this. So we have our domain, our kingdom, phylum, class, order, family, genus, and species. So basically all of these can be used to identify and classify an animal. And the way to remember these levels is with the saying, "Dear King Philip came over for green soup." So domain, kingdom, phylum, class, order, family, genus, species. So you can use this to remember the order. Sorry, it keeps doing that. Of taxonomic classification of animals or plants or any other organism. All right, and then going into, sorry, my computer just keeps disconnecting from the iPad, so please bear with me. So moving on to phylogeny. So phylogeny is the history of evolution of a species or a group. So basically you're looking at the lines of descent and relationships. So here I have a phylogenetic tree, when it comes back. Phylogenetic tree showing the relationship between bacteria, archaea, and eukarya. And you can see that they all lead down to a common ancestor. So basically by using phylogeny, we can relate all of these different organisms and eventually we can find where they have a common ancestor of some sort. The most accepted method to construct these phylogenetic trees, one of the best methods to do it is using cladograms or cladistics. So this is when you're looking at the measurable characteristics that organisms have in common. So basically in the assignment that you did, you had to create cladograms. So you're looking at different traits that animals had and relating them in that way. So in this case, you can see that at the very bottom, we have the hagfish and then you have a perch. And so the difference between these two organisms is that the perch has a jaw and the hagfish does not. But as you move up the cladogram, each of those organisms are going to have jaws. So eventually at each node, which is each of these points at the end, like where the two lines meet, that's where they would have a common ancestor of some sort. So this is one way to construct a phylogenetic tree. This is the most accepted way, probably the easiest way to do it, because in phylogeny, in phylogenetic trees, other ways to do it, you can use RNA, you can use just different methods, but cladograms are the easiest way to understand. And then briefly going into homologous versus analogous traits. So homologous traits, they're inherited by two different organisms by a common ancestor. So in the case of human hands or a cat's paws, like we have similar bone structures, and this is because we inherited these by a common ancestor. And eventually over time, like humans, horses, cats, bats, all of these different organisms, we all inherited different traits. We all evolved to have these same structures, but they are different because of our needs, if that makes sense. So that's homologous traits. And then analogous traits, there's similarity due to convergent evolution. So it's not from a common ancestor, and they're independently evolved, even though these traits are somewhat similar. So here I have a few comparison of analogous structures. So in the case of an insect leg and a horse leg, even though they both have legs, they're not essentially, they didn't necessarily get their legs because of a common ancestor. They evolved with those on their own. Same with a fish fin and a dolphin flipper. Dolphins are not fish, they're mammals. So they didn't evolve that trait because of their common ancestor. They evolved that trait because of their environment. And the same goes for insects and birds. Even though insects have wings and birds have wings, there's a lot of stark differences between the two. And yeah, they evolved wings separately in their environments. So that is chapter 11. I know we're just kind of breezing through this. Are there any questions before going on to module 12? Okay, so now we'll move on to module 12. So this is the one that we're working on right now. So describe the uses of prokaryotes in food processing and bioremediation, examine the endosymbiotic theory, explain the origin of mitochondria and chloroplasts, describe the main characteristics of protists and their roles, and summarize the importance of fungi in the environment, food, beverage preparation, so on and so forth. So going on to prokaryotes, something that I would recommend that you review before, oh, sorry, I just saw the question in the chat. How far can a common ancestor be to be considered one? That's a great question. If you're, that is a great question. Sorry. So if you're basically looking at, in this case of the cladogram, you're just looking at different traits and relating them. So we know at these nodes where the two lines meet, there is a common ancestor, but we don't necessarily know what it is. So using this method, we can kind of infer that there was some sort of organism that occurred in between those. I don't think it's necessarily how far. It's just kind of like phylogenies and cladograms are used so that we can kind of break it down and try to find that common ancestor, if that makes sense. Okay, are there any other questions? Okay, so going back to prokaryotes, I've definitely reviewed the difference between prokaryotes and eukaryotes in their cells. So of course, this is having to do with having a nucleus or not having a nucleus, what types of parts in the cell would not be in a prokaryote and would be in a eukaryote. So I definitely just briefly review that. That will be coming up over and over. So it's just good to know that kind of off the top of your head. And going into food processing, prokaryotes and other microbes are beneficial to some food production. So in the case of cheese, bread, yogurt, different prokaryotes can be used in order to produce those products, and they can help transform the texture of that product. They can provide flavors. In some cases, prokaryotes are added so that they can fight off other types of prokaryotes from infecting or contaminating the food product. So I would definitely know that cheese, bread, and yogurt can be created through prokaryotes. So in the case of bread, like yeast can be used. All right, and then going into bioremediation, so this is still talking about prokaryotes. So prokaryotes can be used to remove pollutants such as toxic metals and oils from oil spills. So this is actually one method that they have been using to clean up these big oil spills or clean up runoff from factories that have had leaked things like toxic metals. And so this has been a really effective way in recent times to clean up those areas and help the environment around there without having to use things like plastics and all of this. They just put down prokaryotes and they do all of the work. All right, and then this module also talks a bit about the endosymbiotic theory. So basically, eukaryotic cells may have evolved when multiple cells join together into one. So this is talking about chloroplasts and mitochondria. So chloroplasts, they were existing as a photosynthetic bacteria, whereas mitochondria was a heterotrophic bacteria. So heterotrophic, that basically means that the mitochondria had to get its food from somewhere, whereas with something that's photosynthetic, they create their own foods, much like plants and animals, which we will go on in a little bit. So basically, the endosymbiotic theory, it states that eukaryotes are a product of one prokaryotic cell engulfing another one, living with another, and evolving together over time until the separate cells were no longer recognizable as such. So basically, in the diagram, you can see that this one prokaryotic cell, it met up with an aerobic prokaryote, so that would eventually become your mitochondria. It was engulfed by this prokaryotic cell. And in the case of chloroplasts, chloroplasts existed as a photosynthetic prokaryote on its own that was also engulfed by a cell. And eventually, as evolution went on, these cells would all become one and all would become prokaryotic. All right. And then I'm looking at the chat right now. So we can consider fermentation as prokaryotic food processing. I would say so. Let me make sure. Yeah, so yes, so prokaryotic cells can be fermented. They can use fermentation pathways. So in the case of yeast, alcohols can be made. Yes, so on and so forth. And then, sorry, going into fungi. Fungi, I will like to remind you, are not plants. They are considered eukaryotes, and most of them are non-motile, so that means that they cannot move, like they usually stay in one place. They are used in fermentation in some cases, and they may include molds. So these are types of fungi. These are some of the traits that they have, and I would just know those for the exam. And then also I would know the uses as well. I didn't really get to go into the uses. I ran out of time earlier today, but just know briefly about the different types of ways that fungi can be used in pharmaceuticals, in foods, chemicals, so on and so forth. And going back to protists, basically they're producers in the world's oceans. They can be single multicellular, can live in many different environments, and many of them are motile. So that means that they can move around on their own. All right, So that is module 12, and that is pretty much where you are at right now with all of your modules. So before going ahead, do I have any other questions? Yes, so the endosymbiotic theory, this is basically talking about how eukaryotic cells came to be. Yes, so this is just talking about how the mitochondria and the chloroplast, this is a theory, so they might have existed as prokaryotic cells outside of another prokaryotic cell. They were completely on their own, and then they were engulfed, and eventually the chloroplast, mitochondria inside of the cell all evolved to be just one cell altogether, becoming a eukaryotic cell. Yes. So that is the endosymbiotic theory. Are there any other questions before moving forward? Okay. So going on to module 13, this is the module you're going to work on this upcoming week. So we're going to talk about the adaptations that allow plants to colonize land, challenges involved, compare and contrast characteristics of non-vascular plants and vascular plants, identify features that distinguish the animal kingdom from other kingdoms, explain the main characteristics of different types of organisms, and name and describe the distinguishing features of 3 main groups of mammals. So I'm not going to go into every single point on this module because I want it to be fun. I want it to be something that you discover. But I'm going to go over the main learning objectives that are covered on the exam. So first off, we have our non-vascular plants versus our vascular plants. So with non-vascular plants, these plants have no xylem, they have no phloem. So these are parts of the plants that bring up water and sugar and usually make plants rigid. So when plants do not have that, they are considered non-vascular. And also, I put no lignin. So lignin is a type of chemical that makes the plant rigid. So if a plant is not rigid, it is considered non-vascular. And non-vascular plants, they're also referred to as bryophytes. And some good examples of these would be like moss, and basically anything that does not have a xylem or phloem. So the way that you can remember this, with the xylem and phloem, basically with most of the plants you know of, like flowering plants, when you water a flowering plant, you know, you can water them every other day and they will use their roots and they will suck up that water and they will use it eventually in their photosynthetic processes in order to survive. Non-vascular plants, good example of these are mosses. So with mosses, usually they grow in places that are dark and wet and usually have an abundance of water. You wouldn't typically find mosses living outside, like in the sun, and that's because they can't collect fluids on their own. They don't have that xylem and phloem to draw up the water into them. So non-vascular plants depend on having water in their environment all of the time in order to survive. Oh my gosh. Okay, I hope this stays this time. So sorry about that. So here I have a different comparison of living plant divisions. So you can see that the non-vascular plants, examples, mosses, and then other plants are considered vascular plants because they have a xylem and phloem and lignin. So now briefly going into the animal kingdom. So the animal kingdom, unlike other kingdoms, they're made up of multicellular eukaryotic organisms. So they have a body plan in place, and their diets can vary. So different animals can be herbivores or carnivores or omnivores. Most animals reproduce sexually, not asexually. So the majority would be produced that way, and they are heterotrophic. So before, like I mentioned, if an organism is heterotrophic, they rely on eating in order to get the nutrients that they need to survive. Something that is autotrophic would be considered a plant, because a plant makes all of its sugars on its own just by sitting in the sun through photosynthesis. We can't photosynthesize. We rely on eating things like plants, eating things like other animals in order to survive. So we are heterotrophic, and animals usually are motile, so they can move. And mentioned before with like the fungi usually stay in place. Also plants, they just kind of stay in one spot for their whole life cycle, whereas we are very motile. We are moving constantly. We can walk across a whole field without as much effort as maybe a plant would have. The plant usually stays in one place. So that is the animal kingdom. And then we briefly will be talking about the different types of organisms that are within the animal kingdom. So we have arthropods, amphibians, reptiles, and birds. So arthropods, these are including your insects, arachnids, myriapods. Sorry, I misspelled that. So these are including your millipedes and your centipedes, your crustaceans. So these are your crabs, your lobsters. And basically, arthropods, they have functional segmentation of the body. So you'll notice with insects, they have a head, thorax, abdomen, so they're segmented. They have the presence of jointed appendages. So if you think of ants, if I go back to the picture of this ant, you can see their leg is segmented. And they have an exoskeleton, which is composed of chitin. So that's what you would typically find in arthropods. Amphibians, these are including organisms such as frogs, toads, salamanders, and newts. They live a dual life. So in this case, they go through metamorphosis. They have a stage where they're larvae, and they have a stage where they're adults. So in the case of a frog, we have tadpoles, and then we have, when they're an adult frog, they can come out of the water. So they have 2 different stages to their life. They have 4 well-developed limbs, and they have moist, permeable skin. Going into reptiles, reptiles include crocodiles, snakes, lizards, turtles. They usually have scales, and they are usually cold-blooded, so they are ectotherms. Whereas birds, which have feathers, wings, a lightweight skeleton, and a beak, they are warm-blooded, so they are referred to as endotherms. And similarly, in mammal groups, which include humans and other animals with hair that feed their young with milk, they are also endothermic. They are also warm-blooded. So we are warm-blooded as well. And then lastly, this module briefly talks about different types of diversity and diversity in general. So diversity is the variation of life forms present in different ecosystems. So basically, if you have a certain number of species within your ecosystem, you could have more diversity or less diversity. So if there's too much of one species compared to all of the others, you're going to have less biodiversity. Or if you have a bunch of different species living in an ecosystem, you would have a lot more biodiversity. So one of the most biodiverse places on land, at least, is like the rainforests. So they have a ton of organisms, all different species. Some species we haven't even discovered yet. And so those are considered a lot more biodiverse compared to, let's say, a desert where very few organisms can survive, very few species can survive. So those would be less diverse in comparison. And you go over alpha diversity, beta diversity, and gamma diversity. So I would just know that alpha diversity is a measure of species richness by the number of species in a given community, so that's the local. And species richness is, like I said before, if there's a lot of different type of species in that area and there's not too much of one compared to another. With beta diversity, this is the ratio between local diversity and regional diversity, and gamma diversity, this is the total diversity of a landscape. It's kind of a combination between both alpha and beta. So there's going to be a video that you watch that has a lot of visuals and explains it a lot better, but I would mostly know that about alpha diversity. That's the most common and straightforward one. And yeah, so that's pretty much it for all of the modules. I feel like this exam isn't as detailed, as I mentioned before, as the last exam, because you're not going through all of the stages of mitosis and meiosis. We're basically going into evolution and diversity. So yeah, that's pretty much it for all of the modules. Those are some basic notes. I would highly recommend making a study guide or watching the videos that are with each module and just going through reviewing each learning objectives. And if you can, definitely try to do the project that will be assigned next week. That would help me out a lot. And yeah, I hope you all get the most out of that. And I wish you all the best of luck with your upcoming exam. So if you have any other questions, you can feel free to put them in the chat. I'll kind of be hanging around here for the next hour, so feel free. But thank you for coming and have a good one. Of course, thank you. Oh, and like I mentioned before, I'll be posting the